In this lecture, we're going to talk about metabolism, which is all the chemical reactions that are happening inside the cell. And anytime we talk about chemical reactions, we do always have to talk about energy that's going in to make those chemical reactions occur, or energy coming out from different chemical reactions. And if you recall from chemistry, the chemistry unit, everything consists of matter, stuff, and energy, whether it's in motion or whether it's stored energy. And energy is defined as the capacity to perform work. So it's just, what is needed to do some type of work, whether it's chemical processes or whether based on these pictures it's used to go up or down a hill. So there are various forms of energy, but the two major types that we're going to talk about and focus on are kinetic energy and potential energy. Kinetic energy is the energy of motion or movement. Examples just on that previous slide is the biker going up the hill, the biker going down the hill. Again, those are energy of motion. Whether it's going up or going down, biker is still moving. Whether it's you're driving your car and your car's in motion, we can physically see a lot of this energy in motion, this kinetic energy. There are, however, molecules that you can't see that are going through this energy of motion as well. Just in a glass of water, the water itself, those water molecules that make up your glass of water are constantly moving around. Potential energy is stored energy. So it's not moving. We don't actually see a biker moving anywhere. They're not going up the hill. They're not going down the hill. They do, however, have the potential to move. And so there's stored energy somewhere that allows them to have that potential to whether go up the hill or go down the hill. Now, a lot of times potential energy or stored energy is in the form of chemical energy. When I talk about chemical energy, it's a type of potential energy, it's a type of stored energy, but the energy is stored in chemical bonds, which is where it gets that name chemical energy. So anytime you break a bond, anytime you break something up, it releases energy that's stored in those bonds. An example, a candy bar sitting on a shelf has stored energy in it. You don't get any of the energy till you eat that candy bar, and your digestive system breaks all those chemical bonds that's holding all of those different nutrients in there. I say nutrients loosely, but as it's breaking apart all of those nutrients that are in there, as you break those bonds, it releases energy. Now you get energy from that candy bar. So it's just energy stored in bonds. Now, energy itself can never be created or destroyed, but rather it just changes from one form to another. An example, you can have plants that are going to take energy from the sun, they convert it into food that other animals are going to eat. The plant, the animal, the moose can eat energy that's in that plant, which has chemical or stored energy in it. As it breaks it down, it can use that energy to propel it forward, mechanical energy or this energy of motion. And so you're constantly just going from one form of energy to another form. But if you notice on here, anytime we go from one form of energy to another form, so solar energy to chemical energy to mechanical energy, there is some energy that is lost as heat. So again, it's not destroyed, it's not created, it's just moves to heat energy. So the energy that's in food, some of it helps move the moose forward, but some of it is lost as heat. And the general rule is a rule of 10%, that only 10% of the energy that is in something is actually used or utilized as it goes from one energy type to another. An example, as this moose is eating the plants, the energy that is stored in that plant material is moving the moose forward, but only 10% of the energy that was in that plant is helping move that moose forward, which means 90% of the energy is actually lost as heat. So yes, that would also equate to, of all the food you eat throughout the day, only 10% of it is actually moving you around, walking around, things like that. And 90% of the energy that's in that food is lost as heat. But it does help us maintain our body temperatures as well. But you are constantly exuding heat from your body. Now, there are some laws when we talk about energy. It's not just, well, here's two types and that's all you need to know. There are some laws, and so there are people that study energy and it going from one form to the next. The study of the energy going from one form to another is called thermodynamics. So it's transforming from solar energy. It can eventually go into plants, and through photosynthesis, you can get sugar out of the plants. And again, it's changing forms, solar energy into a potential energy, that energy that's stored in those sugar bonds. But we're going to take a look at two laws of thermodynamics, again, as it's going from one form to another. And the first law of thermodynamics, we've already mentioned this idea in this class or earlier, and it's just that energy can be changed from one form to another. We can go from solar energy to potential energy, but energy in that process is not created, it's not destroyed, it's just changing forms. So it's not lost, we don't lose energy, it just changes forms. The second law of thermodynamics is that as we go from one form of energy to the next, we do increase disorder. And so disorder, also known as entropy, happens. Things are just not as organized as we go from one form to the next. And when we do go from one form to the next and we create this disorder, heat is usually released when that energy is transformed. Again, this is just showing, we've got food going into muscles. Muscles, when they contract from walking, running, lifting weights, those contracted muscles are going to generate heat. It's one of the main reasons why you get extra warm when you are working out, is because those contracted muscles are taking one form of energy, food energy, and using it, and you have that heat being lost. So you get extra hot when you work out. Fitness centers usually get quite warm because you have lots of bodies that are exuding and giving off more than normal heat. Now, there are two different kinds of reactions that can occur when we are building things, making things, and both of them are going to have energy in their reactions. So, and there's endergonic reactions, and then the next slide is exergonic reactions. Endergonic reactions means you have chemical reactions that are either going to store or release energy. So endergonic meaning we're going to have energy that is put into the reaction, and this is where you're usually going to have some stored energy. So we're going to put energy in and we can store that energy. Ender or end means in, and so endergonic reaction means energy is going to go in. So if I have a couple reactants, whatever they are, a couple different molecules, I can put energy in, which means now my products actually have more energy than the reactants did. And now we can actually have some stored energy there. The exergonic reaction is the opposite. Ex means out, just like an exit means out. An exergonic reaction means heat is lost, it is given out. And so we can see if the reactants have quite a bit of energy to start with, they can lose energy and the products now have less energy. So they are opposites of each other, because again, we can have chemical reactions that require energy to go in, we can have reactions that are going to release energy. We just talk about the word metabolism. You've probably heard the word metabolism before. When people are saying I have a fast metabolism or a slow metabolism, metabolism itself is just talking about all of these reactions that are happening inside of a cell. So this would include all of the endergonic reactions, all of the exergonic reactions. It's all the reactions that are happening in a cell. If someone says they have a very fast metabolism, it just means that these reactions are happening very quickly and usually very efficiently. A slow metabolism, the reactions may be happening a little more slower. Now, because the reactions are opposite of each other, one takes in energy, one releases energy, a lot of times the cells will couple these two reactions together, so that if we need some product and the reaction to make that product requires energy, we have to get the energy from somewhere. And so we can couple it to an exergonic reaction, one that's giving off energy. So we can use the energy that's given off in an exergonic reaction to power an endergonic reaction because it needs that energy. So we constantly have reactions that are giving off energy, and we're just going to couple it with a reaction that needs that energy. It's a wonderful way and an efficient way to make sure that these reactions are all going to happen. Now, I did talk earlier about energy in a candy bar, this chemical energy that's stored in bonds. And it is. And food itself, we can break it down into kind of 3 groups based on how we measure how much energy is in that particular type of food. A carbohydrate, a protein versus a triglycerides. These are your fats. And gram per gram, fats or triglycerides do have more energy, more bonds in them per gram. More bonds equals more energy, so we can get more energy out. One particular chemical that keeps coming up, and it will continue to come up, is the chemical of ATP. So it is a modified nucleotide, so it's not a nucleotide that's used to help build DNA or RNA, but it is made up of a sugar. It is have a nitrogenous base, adenine, but kind of the one more unique thing to it compared to a nucleotide that would be found in DNA or RNA is that it has these 3 phosphate groups in it, and it's where it gets its name. Adenosine triphosphate is where the ATP stands for. It's those 3 phosphate groups on the end that between each phosphate group, there is a lot of energy stored in that bond. So between each phosphate group, lots of energy in these bonds. And if we need to release that energy because we need to use that energy to power some type of exergonic reaction, to power something, we can break off one of those phosphate groups. We can break a bond and release the energy. So that's what it's showing in here. We can add water. It's a process known as hydrolysis. Just means we're using water to break things up, and we're going to break off that phosphate group. We're going to break that bond. And so we released one of the phosphate groups, and because we broke that bond, we released energy from ATP. And now you have a molecule called adenosine diphosphate because it only has 2 phosphate groups on. So anytime our body needs energy to do things, it uses ATP and just breaks a bond. It breaks one phosphate bond off of that ATP. However, at some point, we need to make and regenerate that ATP. And so as we eat things throughout the day, the energy that is stored in that food is used then to add the phosphate back onto ADP and make and regenerate our ATP. So we're constantly taking ATP, using energy to make using energy from that ATP, and it results as ADP. But we can take energy from food that has stored energy, and we can add that phosphate right back onto ADP, and we're right back to where we need and have that nice potential stored energy in ATP. Adding that 3rd phosphate group on is known as just phosphorylation. So we're adding that 3rd phosphate group on, and it energizes the molecule. It also destabilizes it because it's really easy to break off that 3rd phosphate group, but it's got lots and lots of energy that's just ready to go anytime we need it. So this is showing how we're constantly, regenerating that ATP. So if we have our ATP molecule, there are those 3 phosphate groups at the top, and we're going to break this bond. It's a high energy bond. We're going to break it, and we can release energy. And then we are left with ADP and then this phosphate group. So we can use the energy somewhere else, but now we have these 2 molecules. Well, to go right back to ATP, we can put energy in. So we have this arrow going in, and that's that process of phosphorylation, is adding that 3rd phosphate group on. Does require energy, but we're right back to having ATP again to power and use wherever else we need it. This picture was shown on that video on where we have reactants and we have products. This particular reactant has more energy than the product, so it's going to give off energy because our reactants have more, the products have less. So the energy is going to go somewhere else. It's going to give off energy. Because a reaction is going to give off energy, You wouldn't think it would still need energy because the goal of it is to give off energy. But all reactions do require a little bit of energy just to get them going. The main reason for that is we don't want reactions to occur when we don't want reactions to occur. So to get any reaction to happen, we have to add a little bit of energy just to get that reaction started. And then, although we're going to put energy in, we're going to get a whole bunch of energy out that's going to get released. So the amount of energy that's needed to just get a reaction going is called the energy of activation. So it's just how much energy is needed to get the reaction going. Now, because there are some reactions that require quite a bit of energy just to get them started, that reaction may not occur just because so much energy is needed just to get it started, even if you're going to get more energy out by the end by creating those products. So what our bodies try to do to try to help with that is that we use enzymes, because enzymes, the presence of enzymes, lower how much energy is needed to get that reaction started. So they lower the energy of activation. So this is showing how much energy, this energy of activation, is needed. It's quite a bit just to get the reaction started. And this is without an enzyme. However, if we add an enzyme, the amount of energy that's needed to get the reaction going is only this much. And if less energy is needed to start it, it's more likely to happen. I like this analogy with the frogs jumping over this big, huge barrier. So if these are my reactants and these are my product frogs, for these reactant frogs to become a product, they have to jump pretty high. They need quite a bit of energy to jump over that wall to become a product. And if they don't have enough energy present, they're not going to be able to jump over that wall and become a product. However, if there's an enzyme present and they lower that barrier, they lower how much energy is needed to become a product, it's more likely that they're going to be able to jump over that little wall and become a product. So enzymes are really, really important in making and ensuring that these reactions are going to happen inside of our cells. So what are enzymes? Well, enzymes are proteins, usually. Sometimes they are RNA, but they're proteins, and their main job is to decrease that energy of activation. So they decrease how much energy is required. Now, they are specific for a substrate. So whatever they work on, work with, they are very specific for what will fit there. This is showing an example of an enzyme, and the only thing that can fit there is this particular shaped substrate. So they're very specific. The active site is where that substrate is going to fit. So it's active, it's where things are actually happening. Now, in any type of reaction where enzymes are present, the enzyme, even at the end of the chemical reaction, the enzyme's not used up, it's not changed, it's just constantly recycled. So the enzyme is going to take this substrate, it's going to fit in there, and ultimately it's going to break it apart into these products. But the enzyme at the end wasn't used up, it wasn't changed, it's just recycled, it's ready to go to find another substrate. Most enzymes end with ACE, and a few end with SYN. Just some examples, there's an enzyme called lactase. For those that are lactose intolerant, lactase is the enzyme that helps break down lactose. So it's very specific. Lactase is very specific for the substrate lactose. Another example of an enzyme that ends in SYN, there's one called pepsin. It helps break down proteins in our digestive tract. So most end in ACE, and a lot of times even their names tell you a little bit about what their substrate is, but there are a few that end in SYN as well. Enzymes, as I said, they're going to take a substrate and they're going to make a product. Again, they are not used up, they are just recycled, they're not changed. So the two different types of reactions that an enzyme can do, it really is they can break things down or they can build things up. So they're going to take a substrate and they're going to break it down, or they're going to take substrates and they're going to build something with it. So the reaction called degradation, just based on its name, is breaking things down. So this enzyme is going to work and fit with this particular blue substrate. When that substrate sits in the active site, that enzyme's job is to break it apart. And so its product are two smaller products that came from that original substrate. Synthesis is it in reverse. It's going to take two smaller substrates that are going to fit into the enzyme, and the product is going to be one larger product. So degradation is breaking things down. Synthesis, just based on its name, is building things up. This is showing just an example of the two almost working together. So if we have our original substrate is this big orange blob, it's going to fit inside the enzyme right there at that active site. And although the enzyme will leave and it just gets recycled to do something else, your products, it broke something down. So this is degradation. It took this big orange blob, one single orange blob, and it made these two green and purple products. Now in a synthesis reaction, we could take that green and purple, now a substrate, but they were a product here. Now they're going to be our substrates. They can go in, they can fit in an enzyme, and that enzyme is going to put those two things together. It's going to join them together, and it's going to make one larger product. Now, if you've noticed, the enzymes do look similar to each other, but they are different enzymes. So one enzyme is particular for breaking the things down, one is for building them up. But there are things that can affect how well an enzyme works. So enzyme activity is affected by temperature, because temperature can denature proteins. They are proteins. They can change the active site where things can bind. Salt concentration can really affect how things bond and hold together even at that active site. And even pH can affect how well an enzyme works. And most enzymes, especially ones in our body, work best at around a neutral pH. Not all of them. Pepsin that I mentioned before can actually work at a very low pH because it helps break down proteins in the stomach. So it will work very well in the stomach. So every enzyme does have an optimal temperature, an optimal salt concentration, and an optimal pH for its activity, usually based on where it is in the body. But most usually like to hang out kind of in that neutral body temperature, body salt concentration, and a neutral pH. But there are things that can inhibit how well that enzyme will work. So it's not just the temperature, the salt, and the pH. There can also be some things around that can just inhibit them. So an actual chemical or an actual substrate, something else that can inhibit how well that enzyme is going to work. So this is our normal enzyme, and this is the active site where the substrate will normally bind, and it will do whatever it needs to do with it. However, in the body, there are things called competitive inhibitors. It's not the same thing as this big, huge, dark blue substrate, but it's smaller and it's able to fit into this active site. It's not a perfect fit because it's not the original substrate that's supposed to fit there, but instead it fits in that active site, and if it's there, the substrate can't fit there. It's kind of like someone got there first. And so it's called a competitive inhibitor because it's two different molecules that are competing for that one active site to sit in. And if these inhibitors are around, the substrate can't bind, can't make the product. There's another type of inhibitor called a non-competitive inhibitor. And it's called non-competitive because it's not competing for that active space. Instead, it is an inhibitor that binds somewhere else on the enzyme. So there is this little notch on this particular enzyme, and this will bind in this little notch. And when this particular inhibitor binds in this notch, the active site changes shape. If you notice, it was kind of this nice big, almost open W on the left-hand side, but once that non-competitive inhibitor bound, it changed shape. It almost started to fold inward a little bit at the top. And because it changed shape enough, the substrate can't fit in there anymore. And so if the substrate can't fit in, it can't do whatever that enzyme is supposed to do. So it's called non-competitive because it's not competing for the active site space, but it's still an inhibitor because it's still going to inhibit the substrate from being broken down or making whatever it is that it's supposed to. And unfortunately, some of these enzyme inhibitors can actually spell death. So there are some inhibitors that inhibit an enzyme that's irreversible, which means the enzyme can no longer ever make whatever products it's supposed to. Examples of these: cyanide inhibits the enzymes that make ATP. Well, we already talked about it in this lecture, ATP is needed constantly as that quick energy to power any of our chemical reactions by removing that third phosphate. If we can't ever make ATP, our chemical reactions can't happen in our cells, and it's ultimately going to spell death. Serine inhibits an enzyme located at the neuromuscular junction, which means this is that area where you have nerves telling your muscles to do things. And if your muscles can't do anything, whether it's just even the diaphragm, which helps in your body breathe in and out, if your nervous system can't tell your diaphragm to contract, you're ultimately going to die. Warfarin, which you may have heard of, is an enzyme that's responsible for that inhibits the blood clotting process. And actually, you may have heard of this before because there are people that do take warfarin to prevent blood clotting for various reasons. But my note on that, it is taken at a very low dose, and it's very, very regulated to make sure not enough is taken, because if too much warfarin is taken, it could inhibit all blood clotting, which means if there is injury, the body can't clot fast enough. And so enzymes are extremely important in different types of chemical reactions.